Good evening all. Welcome back to brilliant YouTube live classes. In our yesterday's session, we discussed 3 chapters, high weightage 3 chapters. Today's session, in this class also 4 chapters we are going to cover. I hope students, you all are practicing at home because only one day in our hands. Tomorrow only the one day in our hand, next day exam only. So all of you carefully prepare high weightage chapters first, like molecular, biotechnology, principles of inheritance, biotechnology applications. Okay, so today 2 important chapters, biotechnology application. Along with these two more chapters, human reproduction, sexual reproduction in plant chapter. This 4th chapter in this 2.5 hours we are going to cover. So all of you, I am requesting you all take pen and paper and write down these important concepts. Okay, look at here. The first question we are going, and before first question, in sexual reproduction in flowering plant chapter, what all the important concepts are there? What all the important concepts you should cover before the exam? Look at here. In sexual reproduction in flowering plant, first you should cover, student, keep in mind, anther structure, ovule structure, structure of ovule, structure of what they call anther, microsporangium structure, megasporangium structure. What is the technical name of ovule? Megasporangium, right? Inside the megasporangium, megaspore develop. So remember, structure of anther first and the structure of ovule finish. Tapetum, middle layer, epidermis, endothecium, this all finish first. Am I clear? Next, after this one, microsporogenesis, megasporogenesis. This is a very important concept. Remember, formation of microspores, formation of megaspore. Am I clear? This is in anther and also in ovule. Finish that one. After that, pollination, autogamy, geitonogamy and xenogamy. Today I will explain you this concept in detail because lot of questions are coming from here. Monoecious plant, dioecious plant, autogamy, geitonogamy, cleistogamous, chasmogamous flowers also. Now, water pollination, wind pollination, water pollination also important. We will do that one also. Artificial hybridization, simple concept, but this is very important. You should cover the artificial hybridization also. Next, fertilization, double fertilization, one of the important concept. Fertilization, double fertilization, endosperm formation, endospermic seeds, non-endospermic seed. Remember. Next one, endospermic, non-endospermic seed. Last one is apomixis and polyembryony, citrus fruits and parthenocarpic fruits. These are the topic you should finish before tomorrow. Am I clear? Today only after my class finish all this topic. Look at the first question. First question from sexual reproduction in flowering plant chapter. Draw a diagrammatic sectional view of mature and anatropous ovule. They are asking. What is anatropous ovule? Students, remember, if chalazal end and micropylar end are on the same axis, we call the anatropous ovule. Let me draw the diagram. Look at here. Here we know this is style, stigma, and this is ovary. This is called pistil, right? This is called pistil. Now look at here. I am taking here placenta. This area I am taking placenta. Ovule attached to ovary, what they call, there is a placenta. Placentation we learn in the 11th grade also, axial placentation, marginal placentation, right? Placenta. Look at here carefully here. Anatropous ovule I am drawing like this. This is the structure of anatropous ovule. This is funicle. There is a particular area where membranes are not covered. We can say micropylar end. How many membranes are there? 2 membranes are there. Right? So this complete structure is called ovule or megasporangium. What type of end is this? Chalazal end. This is called chalazal end. And what type of end is this? Micropylar end. This is micropyle. Am I clear? Now look at here. When micropylar end and chalazal end is on the same axis, such ovule is called anatropous ovule. Keep in mind, anatropous ovule. So this is anatropous ovule. Go back. Draw a diagrammatic sectional view of mature anatropous ovule. This is the anatropous ovule. This part is called placenta. This part is called funicle, and there is a particular area is called, we call hilum. This is called hilum. Right, student, are you following me? Here, based on this diagram, lot of questions can come in the CBSE. Look at the first question. What is the first question they are asking here? That develop into seed core they are asking. What is developed into seed coat? Go back here. Remember, here if you look at this diagram, there are outer membrane is there. This is the outer integume and inner integume. Outer membrane and this is inner membrane. Outer membrane called testa. Remember, second one inner membrane called tegmen. Can any one of you tell me what is the fate of these two membranes after fertilization? Any one of you? Pavitra, Sania, Kirtraj, any one of you tell me what is the fate of these two membrane after fertilization? After fertilization, these two membrane will become, any one of you? Yes, absolutely, seed coat. These two membranes will become seed coat. Remember, I make clear. Go back to the question. What develop into seed coat? Our answer we got integumes. What are integume? Outer integume and inner integume. I make clear, right? Absolutely. Go to the second question. What develops into an embryo after fertilization? What is developed into embryo? Go here. Look at here. This one. When we talk, exactly in the middle there is embryo sac. This is the embryo sac is there. This embryo sac will become embryo after fertilization, because in the embryo sac only female gamete is there. Am I clear? Now look at the next one. Look at here. Can any one of you tell me that develops into endosperm and albuminous seed? Any one of you answer this one. Which one is going to become an endosperm? Which one is going to become endosperm? Anyone? Sania? Anyone? Ovule? Okay, now think it is not correct. Not ovule, Pavitra. What is going to become an endosperm? Central cell. Absolutely correct. Look at here, central cell. If you take exactly in the middle, there is a central cell is there. This is called a central cell. This central cell after double fertilization, 2n plus n equal to 3n, endosperm formation. Endosperm formation after double fertilization is central cell. Remember, so it is triploid in nature. Go back to the previous question. Now, through which pollen tube entered into the embryo sac? Students, remember, pollen tube entry into the embryo sac is three method. One is called, look at here, if pollen tube is going through chalaza, I can write what? Tell me, chalazogamy. But this is a very rare. Pollen tube commonly go through any one of, very good, absolutely, micropylar end. So this micropylar end is called porogamy. Everyone mark this one. One is called chalazogamy, one is called porogamy. Third one is integume through it will go. When integume through it will go, mesogamy. Remember, mesogamy. But our answer is what? Porogamy. Through micropyle it will enter. Through micropyle it will enter. Right, fine, very good. Now last one. Fifth one is called that attached to the ovule placenta. I will be silent. You have to answer the fifth one. What is attached to the placenta? Look at here. That attach the ovule to placenta. Remember, all of you, look at here, student. If I take this ovule structure, look at here. This is the ovule structure. This is placenta. This is ovule structure, anatropous ovule structure, right? Look at here. This is called funicle. Exactly here there is a hilum is present. So what is attached to placenta? What is attached to the placenta? Very good, funicle. Absolutely correct. I know this chapter is very simple, but we will cover all the points. Remember, these two chapters, student, sexual reproduction in flowering plant and human reproduction, these two chapter diagrammatic question we can expect. I am really telling you, I am sure diagrammatic questions will come, student, especially ovule structure, anther structure, developing monocot embryo and dicot embryo, seeds also. They will come. They will ask. So answer is what? Funicle. Now I am writing what here? It is funicle. Right? Done. Last one. What is the last one? Describe the characteristic feature of wind pollination. But wait. Here it is given what? Albuminous seeds. My dear children, albuminous seeds, non-albuminous seed. I will tell you one seed name. It is this endospermic or non-endospermic. Tell me. Castor. Castor is endospermic or non-endospermic? Any one of you? Castor is endospermic or non-endospermic? Loyal ma'am and Kidraj, Ganga. Castor is endosperm or non-endosperm? I can tell the castor is, Yes, endospermic rat. P gram is what? Non-endospermic. Remember, P gram orchids is a non-endospermic. Remember, all of you. So here it is. Describe the characteristic feature of wind. Wait. My dear student, wind pollination characteristic feature and insect pollination characteristic feature, carefully prepare. Wind pollination characteristic feature are pollen grains are non-sticky, flowers are non-vector, so that means vectorless, flowers are small, flowers are not producing fragrance. Am I clear? And exposed anther, exposed stigma, feathery stigma, sticky stigma, wing pollen grains. This all the characteristic feature of what? Tell me, wind pollination only. Remember, large number of producing what? Tell me, pollen grains. Am I clear? This all the characteristic feature of wind pollination. Remember. Next question. Look at the next question. What is the next question is given? Next question. How does the pollen mother cell develop into a mature pollen grain? They are asking. How is mature pollen grain? How is developed into? How pollen mother cell PMC develop into mother cell? Everyone look at here, student. This is called microsporogenesis process. If I take, let me take here. This is mother cell. PMC. It is also called MMC. I have a question. Can any one of you tell me? PMC or MMCs are actually developed from a particular one tissue is there. PMC and MMCs are actually developed dash. Shall I say sporogenous tissue? Shall I say sporogenous tissue, student, right? Sporogenous tissue is haploid or diploid? Sir, it is diploid. Where it is present? In the microspore sporangium. So I am writing what here? PMC and MMC. This PMC or MMC undergo meiosis division to form tetrad. Am I clear? There are three type of tetrad. Isobilateral. Next one is called what? What they call? Tetrahedral, isobilateral, F shape, linear shape. So this is the tetrad. I am writing. Look at it. This is tetrad. Am I clear? And this tetrad, this is called microspore tetrad. This microspore tetrad eventually give 4 microspores. Haploid, here also haploid, here. This all are called what? Tell me, microspore. Microspores are nothing but male gametophytes or pollen grains. They are asking what? How does pollen mother develop into a mature pollen grain? Illustrate the stages with the label diagram. You have to draw in sequence manner. I have a question. This is one pollen anther. Just assume, student, one of the microsporangium has 100 PMC. One of the microsporangium has microsporangium. Sporangium has 100 PMC. Can you tell me the total number of microspore produced from this anther? Total number of microspore produced from this anther. Any one of you? 100 PMC in microsporangium of dilobe or dithecous anther or bilobe anther. I am writing. This is bilobe anther. Can you please tell me the total number of? No, Keetraj, think properly. Yes, absolutely. 1600 is the right answer. Sir, how 1600? Bilobe anther is tetrasporangiate, student. I think you forgot this one. It's a basic question. Tetrasporangiate. I am talking about the one sporangium only. In one sporangium, 100 PMC. 100 PMC will give what? 100 into 4. That is 400 into 4. Because 4 are there, right? 4 microsporangiums are there, right? Do not get confused. I make clear? So microsporogenesis process, you should write like this. Next one is called what? Describe the structure of mature pollen. Wait. My dear student, you will get this diagrammatic question. Open NCERT. In the NCERT, when the developmental process is there, in that developmental process, initially they given the vacuoles in the cell. In the cell, there is a given vacuole. Look at here. Let me take here. This is a first cell is given nucleus, and here they given the vacuoles are given like this, right? This microspore, how many of you remember, undergo one unequal mitotic division to produce one male gamete, one generative cell and one vegetative cell. Yes or no? One unequal mitotic division, one generative cell and one vegetative cell. How is vegetative cell? Bigger, dense cytoplasm, abundant food materials are there, and generative cell is spindle shape and it is floating small cell. And everyone keep in mind, this surround around the microspore, intine, first plasma membrane, after that intine made up of cellulose pectin, after that 1 minute. Students, carefully look at here. If you read NCERT, it is given intine is continuous membrane, no gap. Intine is continuous membrane, intine made up of cellulose pectin, but exine is the outer layer which is a discontinuous membrane. Look at here. Let me take the complete structure here. If I take the complete structure, outer one is plasma, this is plasma membrane, and outside of plasma membrane what is there? Intine is there. Intine is a continuous membrane, and after this one what is there? Irregularly exine is there like this. Exine is there. Remember all of you. Now here there is a floating cell wall, generative cell, and the bigger cell called vegetative cell. 1 minute, student. Look at the diagram of microspore. How many germ pores I given here? How many germ pores? This is one germ pore, this is one germ pore, and this is one germ pore. If 3 germ pores are there, if 3 germ pores are there, we can say triporate. Triporate. Remember, mark this one. Triporate is a characteristic feature of dicot plants. Triporate pollen grain is a characteristic feature of dicot. Uniporate. Uniporate means single germ pore in grasses. That means monocots. Okay, and remember, exine made up of sporopollen, world resistant chemical. Exine, sporopollen secreted by. Sporopollen secreted by. I'm looking your comments. Sporopollen secreted by. Who secretes sporopollen? Anyone? Ahmad, Yasir, Ganga, Kitraj, Ian. Yes, tapetum. Tapetum is the innermost membrane of anther. Remember all of you, right? Tapetum. Now, describe the structure of mature and pollen grain and draw the diagram, and it show the 2 cell stage. I have a question. What percentage of angiosperms are shaded 2 cell stage? What percentage of angiosperms, tell me? Yes sir, here it is. 60% of angiosperms are shaded at what? 2 cell stage. And the rest of 40% of pollen grains are shaded 3 cell stage. What are the 3 cells? 2 male gamete, 1 what they call generative cell. 1 vegetative cell and 2 male gametes. 1 minute. I have a question. Can you please tell me how many total mitotic division takes place in pollen grain? How many total mitotic division takes place in pollen grain? Total mitotic division. 1 is, any one of you, Pavitra, Ketra, any one of you, Hafiz, how many total mitotic division? Yes, absolutely. 1 is asymmetrical, 1 is what they call unequal, 1 is a smaller and 1 is a bigger. Can you please tell me 60% of pollen grains are released at 2 cell stage? Now, can you please tell me, when will the second mitotic division finish? When will second mitotic division finish? Sir, once pollen reach on stigma, one pollen-pistil interaction done, generative cell undergo second mitotic division to form 2 male gamete. Remember, mark my words, all of you. Done? Clear? Everything clear? Next question. How does megaspore mother cell develop into 7-cell, 8-nucleated embryo sac? They're asking. Look at here, student. Look at here. All of you careful. Look at here. We have seen. Let me take the embryo here. Let me take the ovule. I'm taking. This is a normal, simple anatropous ovule. I'm writing. I can also draw like this. Look at here. This is anatropous ovule. These are called external and internal integums are there. Can you please, any one of you tell me, here there is a diploidal tissue present which is rich in nutritive material. What is this diploid? What is this diploidal one? Nucellus. This is called a nucellus, right? Nucellus. Nucellus is 2n. Can you please tell me, nucellus is homologous to which tissue? Nucellus homologous to sporogenous. Remember, sporogenous also diploid. That is in the microsporangium. This is in megaspore. In megasporangium. Yes or no? This is completely ovule. Ovule, other name is called what? Megasporangium, right? Now what happened? One of the nucellular cell which is close to micropylar end, this is chalazal end, micropylar end. This cell undergo maturation to form a MMC. What is MMC full form? Megaspore mother cell. This megaspore mother cell, again, it undergo, tell me what? Meiosis division. This meiosis division to form linear shape of 1, 2, 3, 4 cells form. This is called megaspore tetrad. Megaspore tetrad. What happened this megaspore tetrad later? Out of the 4, 3 cells degenerate. These 3 cells degenerate and this will become functional embryo sac. I am not completing that total mechanism. You know this one. So directly I am writing what? Embryo sac. That means, can you please answer me? I need 100 embryo sacs. Can you please tell me how many meiosis divisions I need? I need 100 embryo sacs. How many meiosis divisions required? Any one of you? I need 100 embryo sacs. How many meiosis divisions it require? Answer. You should not delay this one. You should answer this one. Come on, any one of you. 100 embryo sac formation. How many? No, Pavitra, think. What I'm asking. 100 embryo sac formation. No, Vignesh. Now, what is this? This Vignesh. No. 100. Look at here. One meiosis will give one embryo sac. Student, don't get confused. This type of question can come. If this type of question can come, your answer will be wrong. No, you should not be what they call lost the mark. Look at here. Out of the 4 haploidal cells, 3 degenerate or 1 only will become functional, right? So for that purpose, can you please answer me? 100 embryo sac formation, 100 meiosis. Now listen. 100 seed zygote formation. 100 zygote formation. So tell me 100 zygote formation. Sir, you need 100 eggs. That means 100 embryo sac and 100 pollen grain. 100 pollen grain. Can you please answer me how many total meiosis division required for me? For the 100 zygote formation, for the 100 seed formation, how many? Yes, absolutely correct. Right? Very good, Sarim. Who is this? No, this is here 100 egg formation, 100 meiosis plus 100 pollen grain formation. I need 25 only. How many of you catch my point? 25 only I need. Why? So 25 into 4 because all 4 will become microspores there. No, none of them are going to be degenerate there. So 4, it is become 100. Am I clear? So 25 plus 100, 125. This type of questions can, remember all of you. Good, very good. Now, draw the label diagram of matured embryo sac. Matured embryo sac diagram, properly prepare and go. This is embryo sac. Let me draw properly here. This is female gametophyte or embryo sac, chalazal end, micropylar end. I am writing these are the antipodals, haploid or diploid, haploid antipodals, central cell, and here it is synergids are there. Synergids, inside the synergid there is a small structure called what? Filiform aperture. This filiform aperture function is guiding pollen tube into synergids. Remember, this all topics very simple. Next question. What is mean by pollination? Transfer of pollen grain from anther to stigma. So here I am taking what? This is anther. From anther, this is stigma. Transfer of pollen from where to where? From this anther to stigma. Who is their mediator? Pollinators are carry this pollen grain. Pollinators are, it could maybe abiotic pollinators, biotic pollinators. In case of abiotic pollinators, wind pollination plus water pollination. Wind pollination plus water pollination. What type of pollination in water hyacinth? What type of pollination in water hyacinth? Any one of you? Water hyacinth and water lily pollination is? Come on, fast answer. Any one of you, fast. Wind or insect pollination? There are water plant, but they are not showing the water pollination. Because in water lily and water hyacinth, the flowers are emerged above the level of water, so that they are showing insect or wind pollination. Water plants are not showing the water pollination in case of water hyacinth and also water lily. Remember. Now, pollination. Explain the structure of germinating pollen grain with the help of label diagram. Oh my God, they are asking what? Germinating pollen grain. Look at here. What is the germinating pollen grain? I can tell here. This is stigma. Pollen-pistil interaction we have seen. Pollen-pistil interaction mean you have to remember something, student. If you read pollen-pistil interaction, you would see there compatibility, self-compatibility, incompatibility. 3 words are there. What is the meaning of compatibility? Let me take first self-incompatibility. That means same species, same plant pollen grain drop on stigma, it reject. No, I don't want it. I will be I'm looking for the new one, new pollen grain. Which pollen grain is that? Same species, other plant pollen grain, genetically different pollen grain it's looking for. That is nothing but cross pollination, right? That's why, students, I'm telling you, if you read the NCERT, outbreeding devices, one topic is there. Finish the topic. Outbreeding devices topic. You know what the complete paragraph is telling? Avoid self-pollination and what they call looking for what? Xenogamy. Looking for cross-pollination. 4 devices are there. One is called dichogamy, maturation of 2 sex organ, that means the antheridium and also archegonium, or you can write that anther or androecium, gynoecium, different interval they matured. Okay, careful, look at here. Now, this is pollen grain drop here. Done. This is pollen grain, and here it is intine is there, right? This is intine and exon also here. Exon also here. And look at here, careful, look at here, what happened, that mechanism. This is called generative cell and this is called vegetative cell. Stigma will secrete mucus and also water, sugars, right? Now these sugars and this water entered into pollen grain through germ pores. Generative cell undergo mitotic division to give 2 male gametes. Done. 2 male gamete produce, pollen tube arise, right? And this pollen tube started to grow. At the tip of the pollen tube, 2 male gametes and vegetative cell will come. At the tip of the pollen tube, 2 male gametes can come. When pollen tube arise, what is the function of germ pores? Through germ pores, pollen tube arise. Which cell will give pollen tube? Vegetative cell. That's why vegetative cell is also called a tube cell. Also called what? Tell me. Tube cell. So once a pollen tube form, if one single pollen tube form, we can say monocyphanogamy. Monocyphanogamy. If it is many pollen tubes are forming, polycyphanogamy, but eventually one pollen tube only can grow towards the embryo sac or ovule. Done. So you have to draw this one and you have to mention this 2 cells means what, and what is this one you have to mention. Done. Next question. Look at here, all of you. Differences between the autogamy, geitonogamy and xenogamy. Before I go to this question, 5 minutes, student. One question is confusing the students always. 1 minute. All of you look at here what I am explaining. Careful, look at here. This is one plant. This is one flower. Look at here, all of you. This is thalamus. Carefully understand 5 minutes. This is thalamus, right? Now this is ovary, style, stigma, ovary. Okay, now these are anthers. Anthers, right? Now here I am writing what? Petals. Male sex organs are there, female sex organ also there, gynoecium also there, androecium also there, right? Androecium also there. So I can call this flower is bisexual flower. Bisexual flower. Done? Bisexual flower. Can you please answer this question? If bisexual flower is present on the plant, look at here, this is, I am writing here, this plant. Here I am representing one plant. Look at here. This is a plant like this. And in this plant, okay, on this plant is what they call, these flowers are there. Shall I call this flower is monoecious? Monoecious. Am I clear? Monoecious, yes or no? Monoecious, that means plant having the both bisexual flowers. Wait a moment. In this plant, what type of pollination is possible? Tell me. Autogamy possible, geitonogamy possible, xenogamy also possible. Yes, yes or no? Autogamy possible, that means same pollen grain drop on the stigma. Next, geitonogamy also possible, that means here if it is taking one more flower is there. For example, here I am writing one more flower. So one flower, anthers are drop on the stigma of different flower of a same plant. Right? So what also possible? Geitonogamy also possible, xenogamy also possible. I have a question. I have a question. Carefully listen here. I am talking about the maize. I am talking about, this is a maize plant. Okay? This is maize plant. Now in the maize, look at it, these are the leaves. Okay, now here this is called the female part is there. This is a female flowers. These are called what? Tell me. Female part. Corn, baby corn. And here they are called, what they call are there, the male parts are there. Here look at here, this is a male parts. That means this is androecium and this is gynoecium. Okay, gynoecium. That means maize plant have male flower separate, female flower separate, but plant is monoecious only. Same plant have male flowers, female flowers. Same plant have male flowers and female flowers. Can you please answer me, in maize plant, what type of pollination is not possible? In maize plant, male flowers are here. This is male flower and this is female flower. Autogamy, geitonogamy, xenogamy. Can you answer this one? Out of these three, which one is not possible? Any one of you, which one is not possible? Sir, it prevents the autogamy. Very good, absolutely correct. Who is this? Absolutely correct. Ayan. Absolutely. Remember why autogamy prevents, sir? Because the flowers are unisexual flowers. This question, this type of question, student carefully prepare. Castor and maize plants are having male flower, female flower separate. So that, let me take here. Here it is only female flowers are there. So where they will go in autogamy? How autogamy takes place? Autogamy not takes place. But geitonogamy is possible. What is the meaning of geitonogamy? This flower, a pollen grain can drop on the stigma of a same plant. So geitonogamy possible. So which all the pollination possible? Geitonogamy possible, xenogamy possible. What not possible? Tell me. There is no possibility of autogamy. One more question, student. One more. I am taking some. Look at here, all of you. I am taking one flower. This is one plant. Careful, look at here. This is one plant, and this plant have only male flowers. This one have only male flowers. Look at here. It has only, look at here, this is thalamus and androecium only present. Okay, and one more plant is here I am taking. This is a one more plant. Okay, and it has only female flowers. Thalamus, okay, only female flowers. This is completely female flower, complete female plant. This is completely male plant. Answer me now. Which all the pollination possible, which all the pollination not possible? Answer me. Male plant separate, female plant separate. I am calling dioecious. Dioecious plants are there. Sir, what is xenogamy is possible, sir? Other all very good, absolutely correct. Xenogamy only possible, no autogamy, no geitonogamy. This point carefully prepare and go student. They can give confusion question in between this one. Monoecious, dioecious. Remember, male, female separate, dioecious. Dioecious. Papaya, watermelon, like this. Male, female, complete separate. Unisexual. If you tell dioecious, male, female in the same plant, hermaphrodite plants, or dioecious, monoecious plants. Monoecious plant, bisexual. No doubt. Look at here. Here, this is very simple definition only. But why I cover the topic? If a question comes, xenogamy is possible in, autogamy is possible then, you can easily write now. Autogamy, self-pollination, definition you know. The pollination, the transfer of pollen grain from anther to the stigma of a same flower of a same plant. Geitonogamy, transfer of pollen grain from one flower anther to the other flower stigma of a same plant. Right? Next, xenogamy, transfer of pollen grain from one flower anther to the other flower stigma of a same species of different plant. Same species of different plant. Look at here. Xenogamy is also called what? Cross pollination. And xenogamy is that one which is genetically, it brings what? Tell me, different pollen grain. Remember, genetically different pollen grain. Sometimes you will get question, which of the following pollination is need pollinator but it is autogamy only? Answer is geitonogamy. Geitonogamy need pollinator but technically it is what? Autogamy only. Look at here. Next question. I told you sexual reproduction and flowering plant chapter diagrammatic questions 100% beta. So careful all of you. What the question is telling? Name the part of mark A in the diagram. B, how does it reach to B? A, sir, it is pollen grain. It reached by the pollinators through pollination process. Simple question. Next, state the importance of C. C is style. What is it? C is pollen tube. I'm sorry, pollen tube. Pollen tube is the one which transfer the male gametes to embryo sac for the pollination or to a ovule for the fertilization. Pollen tube function is to transfer the male gamete to embryo sac for the fertilization or syngamy. Next one, what happened to the part marked D? Where is D? This one is D after fertilization. Any one of you tell me the D option? Any one of you tell me the D option? Shiva and Loyal. Next, who is this? Ayan, you're giving good answers. Can you please tell me what is the future of future of this D after fertilization? Now this one. D after fertilization. Any one of you? It's ovule. Tell me what is the future? What is the fate of this one after fertilization? It will become this one. This one will become what? Now here it is. That means here exactly after zygote they will become embryo, right? Embryo, right? It will become embryo. Yes or no? Embryo inside what? Seeds. Where is seed? Seed is ovule. The entire part will become seed. This entire ovule becomes seed, but this will become what? Tell me, embryo. Am I clear? After fertilization, embryo. Keep in mind. Next question. Draw a diagram of sectional view of monocot seed grain. Mark this type of question. Diagrams questions. What are monocot seed, monocot embryo development, dicot embryo development diagram? Carefully look at here. Okay, now they are asking monocot seed. We know monocot seed is like this. Somehow it is like this, right? I'm talking about the rice. Look at here. This is monocot seed. There is one layer is surround around. This is called seed coat. It is also known as fruit wall, pericarp, fruit wall. Below this one, there is a membrane called alluron. This alluron membrane is endosperm. Alluron membrane. This is called the seed coat or fruit wall. Now here there is a line is there. Yes, we know this one. This one is there. There is a structure is there. Line. Yes or no? Now this one, and here completely this is endosperm is there. Endosperm here. This is an endospermic seed. Here this part is called scutellum. Scutellum. scutellum nothing but cotyledon, single cotyledon, and here these structures are present like this. Okay, these structures are there. This one is called plumule. This one is called what beta? Coleoptile, coleorhiza. Coleoptile out upper side, coleorhiza lower side. Now they are asking, label the part of function of coleoptile, coleorhiza. This is called coleoptile. Okay, lower one, coleoptile, and here this is known as what we can write, plumule. It protect the plumule. Coleoptile protect the plumule. Coleorhiza protective sheet of radicle. Enough. Coleoptile, coleorhiza, and scutellum is a cotyledon, papery, single cotyledon, endospermic seed. This is endospermic seed. Done. Now next one is next what they are asking, coleoptile and endosperm. Endosperm function is what? Endosperm is a nutritive tissue for the developing embryo. Endosperm is a nutritive tissue for the developing embryo. Done. Next question. Human reproduction. Whatever the topics I covered, artificial hybridization I did not cover. Prepare, that is an easy topic only. After this one, polyembryony, parthenocarpy, this all topics you have to prepare. Make clear? I have a short time, that's why I could not cover all the parts. Okay, now human reproduction. One of the important concept, human reproduction contain diagrammatic questions can come. This is a very important chapter. One by one we'll discuss what are the diagrammatic questions come, how many, 5 mark, 3 mark, one by one. Spermatogenesis, oogenesis. Structure of sperm, structure of oocyte. Spermatogenesis process, everyone should be clear before exam. Before exam, primary spermatocyte, secondary spermatocyte, next one is spermatids, and also spermatozoa. Which one is haploid, which one is diploid? You have to be perfect. We will discuss today. Structure of sperm, structure of secondary oocyte, that also you have to very careful. Menstrual, this one is the very important student. Which hormone increasing during the menstrual time, which hormone peak level at the ovulation time, which hormone peak level at the luteal phase, you have to be remembered, because diagrammatical graphical questions are come from this menstrual cycle. Next, fertilization process. Next one is called embryo development process. This embryo development, my dear children, carefully look at here. NCERT diagram, the fallopian tube and the diagram developmental stages are given. Scan the diagram. Carefully look at the diagram. Which stage is given A? Which stage is given B? What is morula? What is blastula? What is zygote stage? Where are trophoectodermal cells? Where are inner cell mass? Thoroughly prepare the diagram, because diagrammatical questions can come from human reproductive system. Hormones of secreted by the placenta, ovary and testis, and structure of Graafian follicle, and parturition and lactation. This parturition, lactation and structure of Graafian follicle easy, but hormones, look at the first question. I will discuss these hormones also. Look at the first question, what is given here. Answer the following questions based on the information given below. Based on the information given below. Given below are stages human reproductive. Write the correct sequence order. There are order is given: insemination, gametogenesis, fertilization, parturition, gestation, implantation. I will be silent for a while. Any one of you do the answer. Any one of you can give this answer. Any one of you? Shiva, can you do this? Any one of you do this? Which one is the first one? Tell me. Which one is the first stage? Insemination, gametogenesis. So shall I call gametogenesis is the first? After that, insemination 2. After that, fertilization 3. Yes, after that fertilization 3. After that, implantation 4. After gestation 5. Parturition 6. Is this process is clear, correct or not? Ashish, right, very absolutely, very good. Who is this? Ashlesh, very good, absolutely. These all are the steps. First one, gametogenesis. Gametogenesis leads to the insemination. Insemination leads to what will be fertilization. After that, implantation and gestation, parturition. Correct, very good. Next, name 2 hormones that can be found only in the blood of pregnant women. Mention the source of organ tissue that's secreted from. Which organ is going to secrete that these 2 hormones exclusively more in the pregnancy women? What is your answer? If this type of question come, chalo, any one of you tell me, sir, these 2 hormones in pregnancy women. Any one of you? Come on. Rahul, Rinku, anyone? Rinku Kumar, can you? LH, no. LH is normally in all females. I'm asking a pregnancy female, pregnant woman. Very good, absolutely. Who is best? Yes, you're really, yes, you're really best. PHPL, right? Good. Number 1, I can write pregnancy women exclusively placenta secrete 2 major hormones during the pregnancy. HCG, next HPL. Am I clear? One is called what? HCG, HPL. HCG, HPL, these 2 are exclusively in where? In the pregnancy women. And what is the source of this one? These 2 come from the placenta. How simple? Yes or no? HP. What is this HCG? Human chorionic gonadotropin hormone. Human placental lactogen hormone. These 2 hormones come from the placenta, the source of this placenta. Remember, not only these 2 hormones, placenta also secretes. Sometimes you will get the question, write the hormone secreted with the placenta. Placenta secrete estrogen, progesterone, HPL, HCG. Remember, my dear student, keep in mind. Yes, correct. Kidra, yes, correct. Now next question. Draw a label diagram of Graafian follicle. Draw a label diagram of Graafian follicle. Students, you have to be very neat. You have to be very clear and neat diagram you should write. You should draw in the exam. Look at the Graafian follicle. Once this this follicle come to a maturation, which hormone will mature? Which hormone is essential for the growth of this follicle? FSH. Once it's attained to the maximum size, okay, this is a Graafian follicle. Graafian follicle surrounded by theca externe, theca interne granulosa cells. These all are granulosa cells. External, internal granulosa cells are there. Remember, look at it. These all are granulosa cells. One more student, remember all of you. In the Graafian follicle, these all are what? Granulosa cells. In Graafian follicle, there is a big cavity form. Antrum, we say. This is called antrum. Remember this part. Antrum, liquor follicle. This antrum. Exactly here, there is a secondary oocyte is present. This is secondary oocyte, and this is called cumulus oophorus. There is a particular layer. Granulosa cells are surrounding around the secondary oocyte, and it will be hang with the mass of cells called cumulus oophorus. This is the structure of a matured Graafian follicle. You can easily recognize this is a mature Graafian follicle with the help of an antrum. Look at it. When the antrum is there, you can easily identify this is, say, Graafian follicle. On this Graafian follicle, 2 hormones. One is called LH hormone, FSH hormone, pituitary hormone act, even ovarian hormone estrogen act. It rupture, ovulation takes place. Secondary oocyte. This is called secondary oocyte release. The process called ovulation. On 14th day average, 14th day takes place. So draw the label diagram. This is called a Graafian follicle. Come to the second one. Write the functions of following. Corpus luteum and acrosome is given. Answer me, any one of you, after ovulation, this remaining of Graafian follicle called what? Corpus luteum. Corpus luteum is the major source of dash hormone. Progesterone hormone, sir. This corpus luteum secrete progesterone hormone. So progesterone hormone maintain the endometrium thickness. That's why after ovulation, progesterone hormone concentration increase. If this egg is not fertilized, progesterone hormone slowly come down. So next 28 after days, menstrual cycle, one more menstrual cycle start. That means egg has been not fertilized. Got my point? Acrosome is the one which is the upper. Look at here. This is the sperm head part. If you take, okay, this is the sperm head part. On the top of this sperm, there is a modified Golgi body known as acrosome. This acrosome is modified Golgi body. Major function of acrosome: it helps to penetrate deep into the egg. It releases enzymes which dissolve the egg membrane. Zona pellucida, corona radiata, and also vitelline membrane will be dissolved by the specific enzyme so that the sperm can penetrate into the secondary oocyte for the fertilization process. Remember, all of you. Done? Till here everything clear? Look at the third one. Explain the process of spermatogenesis with neat diagram. Identify the haploid, diploid. Now this process I'll explain you. Keep aside. Can you please tell me, during the spermatogenesis process, what are haploid? Which stage of cells are haploid? Any one of you tell me? Any one of you tell me? Come on, Shiva, answer my question. Yes, best. Where are you? Ayan, answer me. Kitraj, answer me. Which are haploid? Which all are haploid? Sir, I can write that secondary spermatocyte cells are haploid. Let me write here. First one is called spermatogonia. Yes, spermatogonia. This spermatogonia is diploid, and this spermatogonia undergo first multiplication, that means mitotic division. And after that new cell, that means these daughter cells form, and these daughter cells are called, what, tell me? These daughter cells are spermatocyte. What type of spermatocyte? Primary spermatocyte. I am not talking about the total mechanism. Spermatocyte. This spermatocyte undergo meiosis 1 to form two, what they call secondary spermatocyte. Secondary spermatocyte finish meiosis 2, yes or no, to form a structures called what? Stages called what? Spermatids. Spermatids are later mature to form functional, that means clear matured spermatozoa. Remember, all of you, which all haploid, which all diploid. Sir, in this total spermatogenesis, primary spermatocyte is diploid, spermatogonia diploid, secondary spermatocyte is haploid, spermatid haploid, spermatozoa also haploid. Remember. So this is the, what they call, mechanism. Can you please tell me here, one spermatocyte, one secondary spermatocyte will give what? Two sperms. Sometimes you will get, in a particular given testis, this many secondary spermatocyte cells are there, this many primary spermatocyte cells are there. Count the number of sperms. One primary spermatocyte will give 4 sperms. Remember, because this is a diploid one. Diploid one, after meiosis, 4 cells will come. From here also 4 cells will come. Right? So remember, spermatogenesis, thoroughly prepare and go. Which one is haploid, which one is diploid, also properly prepare and go. Done my point? Next, look at the diagram. I will be silent. Just assume this question repeated in exam. How can you answer? I will be silent. Come on, all of you. This is the right exam. Like, just you got the same question. Come on, Ayan, answer me. Shiva, and who is this Keetraj? Best. Come on, answer me. You have to answer this one. Read the question carefully. What is this X? The question tells us, I'll not explain. Just I am reading. The hormone X. Label the hormone in the diagram. In this complete diagram, day 7, day 14, 21, and this is the one they are asking, X. Oh, very good. Absolutely. Ayan, absolutely. Very good. Ganga, this is called LH surge. Can you please tell me one more hormone also peak level at this time? One more hormone peak level at this time. One more hormone peak level, Ashwin. Yes, very good. One more hormone peak level at this time. Sir. Yes, LH surge. It's absolutely correct. One more hormone peak level. What is that? FSH, yes, estrogen hormone. FSH hormone, pituitary hormones, ovarian hormone. Carefully read the question. Are they asking about the pituitary hormone peak level or ovarian hormone? If it is the ovarian hormone, estrogen. Pituitary hormone, LH. Right? Very good. So, difference between the morula and blastocyst. My dear children, blastocyst and morula, 2 stages different they are asking. If you are able to recall, morula is 8 cells to 16 cell stage is called morula. Yes or no? Morula does not contain blastocele. There is a blastocyst, blastocele structure is not there. Yes or no? Next, morula you would see at the end of the fallopian tube. At the end of the fallopian tube. But the blastocyst stage you would see in the uterus. If you able to recall theory classes, what we discussed, this is zygote. This zygote undergo, tell me, cleavage patterns to form cells like this. These are called blastomers. So it will divide it into many times, 4 cell stage, and after that 8 cell stage, after that 16 cell stage will form, right? This structure is called morula. Morula. After that what happens? Compaction takes place. Compaction takes place. What is the meaning of compaction here? Smaller cells will be arranged at the lower side, we call trophoectodermal cells, okay, and the larger cells will be arranged in the inner side like this. Larger cells are arranged in the inner side. Okay, slowly what happened, this mass, inner cell mass pushed to one side, so there will be a cavity form called blastula stage. At the time of blastula stage, I have a question, one question. If you answer this, you are properly preparing for the exam. I am sure. Tell me, blastula cyst, morula stage, out of these two, where zona pellucida not there? Out of these 2 stages, where zona pellucida not there? Any one of you answer me? Shiva Kumar and what is this? Muhammad. Out of this, Loyal, can you please answer me? Out of these 2 stages, which stage zona pellucida glycoprotein membrane absent? No, not blastomer. I am asking about the stage. Blastocyst, morula. Out of these 2 stages, one stage there is no zona pellucida. Any one of you? Ganga, are you there? Kirtiraj, can you please answer? Out of these 2, one stage there is no zona pellucida. Come on, answer me. Tomorrow, day after tomorrow our exam, you should be very fast. Come on, answer me. Blastocyst stage, sir. Yes, blastocyst stage. Why there is no zona pellucida? Because the trophoectodermal cells will release the enzymes during the time of implantation. So zona pellucida dissolve. After fertilization, corona radiata will be destroyed, that means degenerate. Zona pellucida will be there. Remember, which of the following egg membranes are persist till implantation? Zona pellucida. Which of the following egg membrane will dissolve after fertilization? Corona radiata. Remember, zona pellucida is a non-cell, it's a glycoproteinaceous membrane. So what are the differences? Morula, 8 to 16 cell stage structure, found in the end part of fallopian tube. There is no blastocele structure, okay. And blastocyst is what? Blastocyst is consists the cavity called blastocele, and the compaction takes place. The inner cell must push to one side, and at the stage of blastocyst, implantation takes place after fertilization 7 days later. That means 7th day after fertilization, implantation takes place in the uterus. Remember my words, students. So diagrammatical question can ask, what is this stage, what is that stage? Wherever implantation takes place, blindly go, that is implantation stage is what? Blastocyst stage. Blastocyst stage. Fix. Next question. Next question. I will be silent. We already discussed this one. Can you please answer this one? Can you please? GnRH, gonadotropin releasing hormone from hypothalamus. This come from the hypothalamus. Hypothalamus, it release what? A and also FSH. What is this A? Shall I call? Very good. Absolutely. Who is this? Moinuddin. LH. Now this is, I am writing, this is LH. LH on leading cell. Leading cells release testosterone, and that is a spermatogenesis. Good. FSH. What is B? What is B? Very good, Shiva. Absolutely good. Sertoli. This is called what? Sertoli. Can you please answer me what is factor D? Sertoli cells are spermiogenesis or spermatogenesis? Come on, answer me. Good. Sertoli cell. Ganga. After this Sertoli, FSH act on Sertoli. Sertoli, that factor. What is this? Spermiogenesis or spermatogenesis? Come on. Abdul. Spermio, releasing of sperms from the seminiferous tubule into where? Epididymis, or we can say vasa efferentia. Spermiogenesis. Okay, this type of question can come, students. Next question. This question, carefully read. You have to give the answer. Just I will read the question only. Which stage is this? Can you tell me the stage name? Morula, zygote, blastula, gastrula. Which stage is this? Which stage? Best. Come on, answer me. Ayan. Very good. Blastocyst. This is called blastocyst, right? Fine. Good. Very good. Blastocyst stage is there. Blastocele is there. Blastocyst. A is what? A is what? Trophoectoderm, right? This is called trophoectoderm. Trophoectoderm is also known as outer layer. Outer layer of this zygote, embryo, trophoectoderm, or we can also write the ectodermal cells. Next question. Look at the question. What the question tells us? Name the stage of human embryo of figure represent. Blastocyst. We already represent, sir. We got one mark. Identify A. We identified, sir. Trophoectoderm. Second mark we got. Mention its function. We did not, sir. Mention the function of trophoectodermal cell. Any one of you please answer me. Trophoblast. Yes, trophoblast cells. Yeah, very good. I forgot this name. Trophoectodermal cell also got trophoblast cells. Trophoblast cells. Absolutely correct. What is the function? Best. Loyal. Ganga, can you please answer the what exactly this trophoectodermal cell function? No, come on. Answer. Sir, it form implantation. It helps in the implantation. Ayan, one is correct. Next, it implantation. One more. Hafiz, one more word is there, but one more function also there. It participates in the implantation. Next, from this trophoectodermal cells only, chorionic villi, placenta develop. Chorionic villi, the finger-like structure deeply penetrate and form what? Placenta. Am I clear? Proliferate, they will be deep present into the where? Embryo, mother endometrium. Am I clear? Remember all of you. So we can write trophoectodermal cells are implantation and slowly it form a structures called chorionic villi. Later they develop into placenta. Next one. Mention the fate of inner cell mass. Oh my God. Inner cell mass fate is what? What is the fate of inner cell mass fate? Sir, inner cell mass are the stem cells. They are the future embryo. These inner cell mass cells undergo the process called what? Gastrulation process. The 3 germ layer form. From this 3 germ layer whole, what will come? Embryo. All organ embryo development takes place. Can you please answer me? In which month genital organ of baby takes place? Which month of pregnancy genital organ of embryo form? That means testes, ovaries, this genital organs, which month they form? Any one of you? Ashwin, Sarim, 6th month, cross check. Ganga, first trimester, second trimester, 6 months. Okay, at the end of the second trimester or the first trimester, at what? Tell me now. 6th, that means at the end of what? 12 weeks. That means what? Tell me. 12 weeks. 12 weeks means what? 4 weeks is what? 1 month. So the third. So here it is. We can write what? Genital organs form first movement, first hair formation, digit formation, heart formation. This paragraph carefully prepare for the MCQ purpose. That paragraph, which month what will develop? Remember. Movement, 5th month. Baby movement takes place at 5th month. Heart, 1st month. So this type of parts carefully prepare and go. Inner cell must become future after implantation of uterus. Inner cell mass will become a future embryo. Where are stem cells located in this embryo? Where are stem cells located? Inner cell mass. Am I clear? Inner cell mass, the stem cells are located. If you are able to recall ADA therapy, gene therapy, today we will discuss exactly this stage. That means early embryonic stage, not this one, before this one. Early embryonic stage we can do this also. Early embryonic stage we can do therapy now, so that the stem cells will become going to full of baby, full of embryo. Right? Next question we can write what? My God, this question, my dear student, I am requesting you all, this seminiferous tubule diagram thoroughly prepare, scan with your eyes. In the brain you should fix beta. If you look at the paper, they draw the seminiferous tubule or the different stages of seminiferous tubule. I don't have time to draw this one, that's why I selected the entire diagram. Draw a sectional view of seminiferous tubule of human, 6 part they're asking, any 6 part. This type of question come. Hafiz, careful, look at here. Amir, Hafiz, Shivakumar, carefully everyone listen here. This diagram 2-3 times practice. Where is the spermatogonia? Look at this is a spermatogonia. After that Sertoli cell. After that primary spermatocyte. After that what? Tell me. Secondary spermatocyte. And after spermatozoa. This process, same manner diagram with the pencil or highlighter properly draw. Am I clear? Seminiferous tubule questions can come. They might give this diagram ABCD. What is ABCD? They can ask. Or they directly ask, draw the seminiferous tubule. Student, human reproduction system, from this chapter we can expect the diagrammatic questions. Right? Now name the pituitary hormones involved. Can you please answer this one? Pituitary hormone involving in this one. Involve the process of spermatogenesis. State their functions. So spermatogenesis process controlled by 2 hormones in case of male. Number 1, testosterone. Number 2, FSH hormone and LH hormone. If you able to recall, previously we discussed one question. This testicular hormone is testosterone. Testosterone is secreted from the testicles. Am I clear? But they are asking pituitary hormone. Pituitary hormone is LH or ICSH. What is ICSH full form? Interstitial cell stimulating hormone. Second one is FSH hormone. FSH acts on Sertoli. Okay. LH act on leading cells. We discussed this one, leading cell. Remember, that's why I'm telling you, student, read the question. Are they asking about the pituitary hormones or ovarian hormones or testis hormones? In case of menstrual cycle, if they are asking if they are asking ovarian hormone, progesterone, estrogen. In case of female, pituitary hormone, FSH, LH. In case of males, testis hormones, androgen, testosterone. Pituitary hormones are FSH, LH. Till here everything? Yes, absolutely correct. Very good. Next question. Next question. Read the question carefully. This is the diagrammatic question from the menstrual cycle. Look at the first question. What exactly they are asking? They are given the different type of follicles growth here given. Okay, different type of follicle. F is what they call, I can add the ovulation, corpus luteum, that etc etc. Identify the following the illustration of a sequence of ovarian events, and 1 to A to 1, a human female they are asking. Identify the figure that illustrate the ovulation and mention the stages of oogenesis it represent. They are asking. Oogenesis, ovulation, shall I call it is F? It is F or not, Sanjay? Look at the diagram. What is the first question answer? Identify the figure that illustrate the ovulation. Sir, ovulation is F. ovulation is F, and mention the stages of oogenesis represent. Mention the stages of oogenesis is A, right? Next, name the above ovarian hormone. Look at here, they are asking ovarian hormone. Read the question, pituitary or ovarian? They are asking ovarian. Name the ovarian hormone and pituitary hormone, both they are asking, both that have caused the above mentioned event. Above this, whatever the actually this is upper diagram, but I given below. Now below given diagram there are two. Ovarian hormone they are asking, pituitary hormone asking. Any one of you help me out? Help me out. Estrogen and LH. Very good. Absolute. Sarim. It's correct. Ovarian hormones are estrogen. Pituitary hormones are LH hormone. Good. Very good. Now, explain the changes that occurred in the uterus simultaneously in anticipation. Explain the changes in the, occur in where? Uterus simultaneously. So, when this process is happening, till this one, this process from here to here, I can write the follicular phase. When follicular phase start in the uterus, the endometrium lining slowly become a regenerating. Slowly become a regenerative, because 1 to 5 days is menstruation due to the low of progesterone hormone, endometrium split. So slowly what on after menstruation, this follicle phase start, endometrium regenerating, and the G after this ovulation, this is called ovulation step. After this one, this is called a secretory phase. When secretory phase come, what happened? Tell me. Progesterone level increase, so endometrium even thicker. Done. Next, write the difference between the C and H. What is C? This is C, growing follicle, and H, this is H. Graphian follicle develop into a corpus luteum. Corpus luteum later convert into corpus albicus. Yes or no? After that, corpus hemorrhagica, and it will be degenerate. So after this one, this is the rest of this corpus luteum and corpus albicans I can write. This is a growing follicle I can write. Am I clear? This is called the growing follicle. C is the growing follicle, maturation which is in the primary follicle, and here it is what, tell me, this is called the corpus albicans, which is the degenerating of what? Corpus luteum. Next, draw the label sketch structure of human ovum prior to fertilization. My dear children, Graphian follicle to ovum difference is there. Look at here. If you carefully look at here, they are asking you to write the secondary oocyte called ovum. This is secondary oocyte. Secondary oocyte have, look at here, this is called plasma membrane. It is also called vitelline membrane, right? And there is a small space also there. This is called perivitelline space. Look at here, this is a perivitelline space is there, right? Now secondary oocyte diagram. After that we can write here, there is a nucleus is one side and abundant food metal are present in the cytoplasm. Abundant food metal in cytoplasm, secondary oocyte, eccentric nucleus, and outside this one we know there is zona pellucida is the membrane, which is non-cellular membrane. Zona pellucida. Remember, all of you, structure you can prepare and go. It is a simple diagram. So this is zona pellucida. Outer side of zona pellucida, there will be a radially arranging corona radiata cells are there. This all are corona radiata granulosa cells are present. So first one is called what? This is corona radiata. This one is called zona pellucida. This one is called perivitelline space. Vitelline space, and this is cytoplasm, we called ooplasm, and this is haploid nucleus. This is called secondary oocyte structure, or I can also write ovum. In the exam, if ovum diagram and Graphian follicle diagram give and ask which one is which one, remember, in Graphian follicle there will be an antrum. But in the secondary oocyte, no antrum, nothing is there. I hope you all are following me. Done. Next question. Look at the next one. This diagram also, look at here. I don't know how many exams you wrote so far in your school. You might have seen this diagram. It is repeating. I will be silent for a while. Can you please answer this one? What is ADEC? Any one of you? Any one of you, come on, Shivakumar, any one of you do this? Any one of you? Sir, ooplasm, yes, okay, ooplasm. A is ooplasm, fine, done. Now what about this one? Study the illustration given and following answer they're asking, right? Now here, identify A, name and state the functions of C. What is C? This one, C is here given, this is D is given, and this is E is given. Can you answer now? What is that one? Right, ooplasm, okay, it is ovum. Ion, this one, they're asking this one, ooplasm, this complete ovum, secondary oocyte of ooplasm or secondary oocyte or ovum you can write. Next one, what? Yes, Devananda, what is this one? Second one is called, what tell me? Name and state the function of C. This is C. What is C? What is C? Zona pellucida. Zona pellucida is the outermost layer of egg, outermost layer of what they call secondary oocyte. And right, next one, function of the C, protecting. Okay, next one is called identify D. What is D? Corona radiata. D is the corona radiata. Done. Next, mention, explain the role of hormones in the formation of releasing of A. Role of hormones. So what is the role of hormones? Estrogen hormone, estrogen hormone, LH hormone, especially estrogen hormone and FSH hormone. No, estrogen hormone, LH hormone, right? They're asking what? Explain the role of hormone which the formation. Yes, formation and releasing. Releasing is called LH, formation is called estrogen plus FSH. FSH. Because follicle stimulating hormone under control only, they started to grow in size. Next, name the part of sperm and which part contain the mitochondria. Wait. Name the part of sperm which having the mitochondria. If you able to recall the sperm structure, we discussed this one in the theory classes. Look at here, the middle portion, middle part is the part which having the completely what is there? Haploidal nucleus, genetic material is there, 23 chromosome, and here it is tail, like this, right? This is a tail is present, and there is a middle, there is a flagellas are present. Now here, this is the exactly area where mitochondria present. Mitochondria present in the middle membrane, we call the nebenken. Look at this is acrosome, middle area. I told you in the principles of inheritance, this mitochondrial genes are not participating in the fertilization. If you able to recall, we discuss fertilization time, the head part of haploid nucleus only entered into the secondary oocyte. Secondary oocyte, remember, middle piece will not go. So these mitochondria are giving energy to the movement of sperm. Enough. Okay, next one. What are the all diagram you should focus? And moreover, oogenesis I did not cover. Oogenesis you have to prepare. Parturition prepare, that's a simple topic only. Next one is called what? Oogenesis and parturition. Female reproductive system also prepare. Look at this one. Female reproductive system, they will give the diagram. They'll ask you A, B, C, what type of what happened in the A, what produced in the B. This type of questions can come. Female reproductive system, male reproductive system, human sperm diagram. Human sperm diagram, carefully prepare and go. Sometimes they directly ask the diagram, draw the diagram, or A, B, C, D they will give. Okay, male reproductive system, female reproductive system. After that, this diagram, ovary, internal structure of ovary. Students, all of you look at here. Primary follicle, blood vessel, tertiary follicle, Graafian follicle, ovum, corpus luteum, everything scan in your brain and go. We should not give chance to a question paper setters to lose the mark. I'm telling you, each and every MCQ, 2 mark, case study, you should rock in the exam. Clear? This diagram, especially this diagram, this one and this two, all these four diagrams from the human reproduction, very important. Especially this development process. Just now I told you, look at that, A, B, C, D, already given here. So careful, look at here, which stage is A, where is D, where is B. Sometimes they give an entire diagram to draw, or they give the diagram, they ask identify A, what happened in A, what is the B. Are you following me? These diagrams are 100% diagrammatic question from where? Human reproductive system. Physics, chemistry and mathematics so far done. They're given a little tricky question. So in biology also, 1 or 2 questions, if they give little tricky, you should ready to face that questions. Let them ask whatever the tricky question they ask. Prepare thoroughly and go. Full confident you can go and write the exam. Next chapter is our favorite chapter. I'll go little slow. I'll cover the maximum question, biotechnology and biotechnology application. RNA mechanism, insulin, 5-mark question from biotechnology application we can expect. There are 3 5-mark questions, student. 1 5-mark question we can expect from biotechnology or biotechnology application. Second 5-mark question we can expect from molecular base of inheritance. Third one can come from any of the other chapters. So biotechnology application, 12 marks according to the CBSE sample paper. So my dear children, carefully look at in biotechnology. Many students are struggling in biotechnology. PBR 3 to 2. I will tell you today which one you should thoroughly prepare and go. Number 1, when biotechnology principle comes, restriction endonuclease enzyme. Restriction endonuclease enzyme, entire history, who discovered, which year they discovered, what exactly this one prepare. Restriction endonuclease enzyme is a defense mechanism of bacteria. Restriction endonuclease enzyme can identify the palindromic sequence and cut at the palindromic sequence of DNA. Restriction endonuclease enzyme called molecular scissors. Biotechnology is possible. RNA technology is possible by the discovery of restriction endonuclease enzyme. Restriction endonuclease enzymes are exclusively found in bacteria. This all point so far 900 restriction endonuclease enzyme have been identified from 230 different type of bacteria. This all information, each restriction endonuclease enzyme, I'm writing ECOR1. I'm writing, look at, this is a genus name, this is the species name of bacteria, this is strain, and this is based on the discovery. First isolated restriction endonuclease enzyme bacteria name. Anyone of you tell me? First isolated restriction endonuclease enzyme bacteria name I am asking. Are you there? I am asking, sir. I have to study chapters. Yes. Can you please answer the bacteria name? It is a Haemophilus yes, absolutely. Ayan, very good. Haemophilus influenzae. Hind D2. So this one is section. After that, construction of RDNA. This is a very, very, very important. Construction of RDNA. Remember, student, 2 are there. Construction of RNA, if they are asking step by step, 1 diagram. Number 2, diagrammatic representation, 2nd diagram. Thoroughly prepare. 2 thoroughly prepare. 5-mark question. Gel electrophoresis. Oh my God, many times gel electrophoresis questions repeated. PBR 3 to 100%. PBR 3 to 2 question sure. PCR, this one also, bioreactor and what they call tools of biotechnology, you can easily understand. My dear students, PCR, PBR 322 and also gel electrophoresis, RDNA technology should not miss. You should not miss. Look at the first question we will cover today. Let me cover this question. Look at here what the question is telling. This is actually from molecular base of inheritance, but accidentally it comes to what biotechnology. Can you do this question, students? I will check how many of you give correct answer. Can you do this? Shiva Kumar, can you do this carefully? Ayan, Shiva, what is given? Fill in the blanks provided. Amino acid phenylalanine valine. DNA code gene AAA CAC. Codon in mRNA what? Codon in, here it is what they are asking. Anticodon what? And here also anticodon what they are asking. Any one of you. Come on, sir. Come on. Fake flip-flop. U, and very good. Look at here. I'm asking mRNA here, this one. So mRNA here it is phenylalanine. This is DNA is AAA is DNA is given. Okay, AAA is given. Template stand. Can you copy here? This is a 5 prime. So U here it is 3 prime. Very good. Absolutely. Flip flop. Where did you go, beta? Correct answer. Next, what about this one? What about this one? Here it is CAC. Okay. Now it is the third 5 prime. I can write GUG 3 prime. Done. GUG. How many have you given? Very good. GUG. Good. Very good, Rohan. Now, what about the anticodon of this one? Sir, UUU anticodon is UUU anticodon, AAA anticodon will come, anticodon or mRNA, right? Very good. Next one is this one. What is this one? GUG anticodon. I will wait for your answer. I will wait for your GUG anticodon. Come on. Sir, GUG anticodon CAC, sir. CAC anticodon in RNA. Am I clear? I'm clear. Till here everything clear? Sir, would you mind explain briefly about it? Yes. Sure, I will explain. I think few students did not get this one. Right, Shiva? No problem, my dear. I will tell you. Look at here. This is a DNA sequence is given here. This is DNA 3 prime. This is 5 prime. 3 prime is AAA is given. Okay, this is what 5 prime. What is the coding strand? Coding strand is TTT, and this is 3 prime. This is a DNA sequence. Carefully look at it. This is DNA sequence. This one is template strand, right? 3 prime to 5 prime template strand, and this one is coding strand. Can you please tell me which of these two will be undergo transcription? Template strand undergo transcription. So I'm writing here template undergo transcription. So 5 prime. So I'm writing here mRNA U because A pairs with U, right? In RNA. A pairs with U in RNA. So first codon is UUU. UUU is a codon. This codon is code for the phenylalanine. Phenylalanine. We know this one, right? Next, UUU. This UUU anticodon is what? Who will recognize this UUU in mRNA? UUU mRNA recognized by the tRNA. So tRNA have the anticodon, right? Now look at here. This tRNA anticodon is A anticodon. Are you following me, bestie? Are you following me? This is anticodon. Same process applied to the second one. What is the second one here? Second one is CAC. Chalo, right here DNA sequence is CAC. 3 prime is CAC. Okay, now this is 5 prime. Write the coding strand. The 5 prime. For coding strand is GTG. This is what? Tell me. 3rd prime. Tell me here. Template strand is undergoing transcription to form mRNA. What is the mRNA will come here? 5 prime. C pairs with GUG. Am I clear? This is a 3 prime. So what is the anticodon? Anticodon mRNA I can write here. It is CAC. This is the anticodon in tRNA. I hope it's clear. I hope it's clear. You understand my point? Clear? Shall I repeat one more time, Shiva? Shall I repeat one more time? Or this type of questions can come. This type of question can come. Remember what is given here in the question. DNA code is in gene is AAA. This is template stand is given. Write the mRNA they are asking. AAA code for the UUU mRNA. AAA code for the UUU. Got my point? Clear? Are you with me? Bestie, Shiva Kumar, are you with me? Ayan, got my point? So this UUU and this is phenylalanine and it is a valine. Now anticodon is we can write AAA anticodon. Here we are writing that anticodon is CAC anticodon. Next question. Now look at here carefully. A polypeptide consists of 14 different amino acids. At least answer this one. Flip flop, where are you? Can you answer this one, Shiva Kumar? A polypeptide consists of 14 different amino acids. How many base pair must be there in the process of mRNA? How many bases are there in the nitrogen bases in the mRNA? Any one of you? Rohan Krishna. Sir, after this, can you please 12 about insertion? Insertion inactivation question is there, my dear. I will discuss entire process. Don't worry. Insertional inactivation, absolutely I'll explain. Don't worry. Now, can you please? Flip-flop, absolutely correct, beta. Absolutely correct. They're giving a 14 different amino acid. One amino acid code for 3 base pair, right? One amino acid code for 3 base pair. Are you with me? One amino acid code for 3 base. One codon is 3 base pair. So one codon is 3 base pair. One amino acid is one codon. So I'm writing what here? 3 into 14. Shivakumar, got my point? Flip-flop, Kitraj, Rohan, you understand my point? What I said? How many? There are 14 base pair. 14 amino acids are there. So nitrogen bases are 14 into 3. 14 into 3. I answer. Sir, what about the start codon and stop codon, and what would be the included? Did they mention the stop codon? They mention amino acids, right? Stop codons are not code for any amino acid. So you should calculate all amino acid codons. Who is this? Rohan? Rohan or Rahan Krishna. Rahan Krishna, in this one, they directly mention the different amino acid, Rahan. Then where is the point of the stop codon? You got my point. Where is the point of stop codon? There is no stop codon here because already 14 amino acids they mentioned. Got my point? Right. Next. That for the code for polypeptide. So 42. I hope you understand, Rahan. Rahan Krishna. Done. Next. How many different type? Answer this one. Answer this one. Flip-flop. Can you answer this one? 14 different type of amino acids are there in mRNA. How many different type of tRNA you need? Very good. Absolutely. You will rock this exam. Sure. Right. 14 different amino acids. Let me take the one example. Few students did not get this point. Look at here. This is 5 prime. 5 prime AUG, AUG, UUU, UUC, GGG. I'm taking. Okay. UAA. Last one is UA and third. This is one mRNA. Students, carefully listen here. This is one mRNA, right? One tRNA is coming. Look at here. One tRNA is coming. This one tRNA, which having the specific anticodon of AUG. What is the anticodon of AUG? It is UAC. UAC. This is methionine. Methionine. Okay, anticodon methionine. Anticodon methionine tRNA will not bring phenylalanine, student. No, not like that. In this class, let me take here. I have one anticodon is UAC. My anticodon is UAC. So I am specific for methionine. Let's take here Rahan. Rahan Krishna have AAA anticodon. He is specific for phenylalanine. So like that, 14 different amino acids are there. 14 different tRNAs required, right? 14 different tRNAs required. Am I clear? Done? Good, very good. So next one. I hope you got this type of higher order thinking question come. Don't panic. Sit properly. What you learn throughout the year, just put on the paper what you learn. Think properly. Think. If this type of questions are difficult, keep aside. At the end, you can try to solve this one. Don't get confused, Rahan. Shivakumar, try to get my full marks in the next exam. Next question. Look at the first question from the biotechnology and biotechnology. Today, I will finish this 2 chapter and I'll leave you. In PBR 322, it is artificial vector. DNA has to be introduced in tetracycline region from the restriction enzymes given below. Which one should be used? Why they are given? Can any one of you answer me? So my mom never taught this. Oh my God! You have to cross this one. You have to practice this. Who is this? Bestie. You have to cross this. You have to practice these questions. Look at here. What the question is given here. This is PBR 322. Okay. This is PBR 322. This is a particular area where tetracycline resistant gene is there, and here there is ampicillin resistant gene is there. ampicillin resistant gene. Carefully look at here. Before I go to this question, any one of you tell me, in the tetracycline place, 2 restriction sites are there. What are they? 2 restriction sites are there. Rohan, sir, how many total tRNAs are there? Many tRNAs are there. Many tRNAs are present, but different type of tRNAs are 61 different type of tRNA for the 61 different type of codon, my dear. 61 different type of tRNAs for the 61 codon, right? Because 61 codons are code for amino acid. Are you with me? You got my point? Who asked this question? How many are there? Rohan, Krishna. 61 different type of tRNAs with the different type of anticodon. They brought different amino acids. But 61 code for 20 amino acids only. Keep in mind, there is no 61 amino acids in the cytoplasm. No, 20 different type of amino acid code for the all proteins that we learn in right now. Now look at here. Tetracycline, there is a 2 genes are present here. 2 restriction sites are there. Can any one of you help me? Can any one of you help me out? PS2, PVU or BAMH1 is there. In the tetracycline, can any one of you tell me in the tetracycline which one is there? BAMH1, yes, flip-flop, right, good beta. BAMH1 plus, one more also there, BAMH1 plus, let me take here, there is, BAMH1 and SAL1, BAMH1 and SAL1, look at here, even I forgot, this is BAMH1 and SAL1 and PV1, PST1, yes, in ampicillin and also tetracycline. Go back to our class question, right, so, BAMH1 is given, right? Now, they are asking BAMH1. These 2 are not there, so answer is BAMH1. Which one should be used? Why? BAMH1. Because BAMH1 restriction site is present in the tetracycline resistant. Then, next, why? Give reason why the other 2 enzyme cannot be. Other 2 enzyme cannot be because these 2 restriction sites are not in the tetracycline gene. So you cannot cut, you cannot insert this one. You got my point? Understand my point? I will tell you one mechanism. Look at here, careful, look at here. Here, if I go here, look at here, this is what here it is, BAMH1 is given. Okay, now go here. Careful, look at here. I have one small question to you all. Look at here, this is, I am writing here, this, look at here, all of you. Now, this is BAMH1. Okay, from here, this is complete, look at here, this is BAMH1, okay. Or I can use this color, BAMH1. Carefully look at here. This is my target DNA. This is my target DNA. Carefully look at here what I am doing. This is my target gene. When I insert this target gene in tetracycline resistant gene here, tetracycline resistant gene here, I constructed rDNA. Can you please, any one of you answer me, when I insert my gene in the tetracycline resistant gene will become inactive or not? Inactive or not, tell me. Tetracycline resistant gene will become inactive because my desirable gene gone inside. Are you with me? Are you with me? Are you with me? Yes, yes. Come on, answer me. If when I insert my desirable gene, but plasmid did not receive. Look at here. This is a one scenario. When I insert gene, plasmid receive. Tetracycline resistant gene will become inactive. When I insert the gene, plasmid did not receive. That means tetracycline gene still working, right? That means tetracycline gene is still working. Yes or no? That means this tetracycline gene is the antibiotic resistant one, can help you to pick up what is your recombinant and eliminate the non-recombinants. Eliminate what? Non-recombinant. That's why I am telling you, student, selectable markers are remember, you can write either antibiotic resistant, biotic resistant, and number 2 is laxy gene. This laxy gene is insertional inactivation. Today 5 minutes thoroughly I'll explain insertional inactivation. Careful listen. So that's why give reason why other enzyme cannot be used. Other enzyme site is not in the tetracycline resistant gene. That's why we cannot use that one. Enough. I hope you got my point. Come to the next question. PCR. PCR question is 100%. Last year also repeated. Okay, I am expecting this time PCR, RT-PCR, reverse transcription polymerase chain reaction, RT-PCR. If you are able to attend, if you are able to recall my MCQ question, I told you there are 2 types of PCR, real-time PCR and reverse transcription PCR. 2 types of PCRs are there. Now listen here. Polymerase chain reaction, Carey and Mills, these 2 amplify the DNA technology. They 2 discover amplification of a DNA technology. This DNA technology, this PCR technique has 3 steps: denaturation, annealing, extension. Come to the question. Identify step A, B and a cycle of polymerase chain reaction given below. What is A? What is B? Only one hint, student. Wherever primers are adding, that is annealing. Absolutely, very good. Absolutely correct. This is a very sharp answer. Whenever primers are adding, I have doubt how many primers. How many primers we are adding? Come on. How many primers adding? Where they are adding? Rohan. Sir, primers we are adding at the opposite of 3rd prime of parent DNA. So 2 primers we are adding. Look at it. This is a 1 primer, 2nd primer. So wherever primer adding, I can write annealing step. Can you please tell me what the temperature there? What the temperature? What's the temperature? Fast answer. So the temperature is 54 to 56 degrees Celsius, right? Now here I am writing 54 degrees Celsius. Next, DNA polymerization and deoxyribonucleate Taq polymerase. Taq polymerase I am adding. So this is extension. B is extension. B is extension. Taq polymerase enzyme. Absolutely, absolutely. Taq polymerase enzyme is that enzyme obtained from the Thermus aquaticus. It is a thermoacidophilus bacteria. This PCR is temperature tolerating what they call reaction. Extension takes place at 72 degrees Celsius. Remember, PCR properly prepare and go, my dear children. Next question. State the specific characteristic feature of an enzyme carrying step B. Step B is extension. Extension takes place in presence of an enzyme called DNA polymerase enzyme. We call the Taq polymerase enzyme. Taq polymerase enzyme is a thermoacidophilus enzyme. It can tolerate at the higher temperature. Got my point? Yes, absolutely. Yes, absolutely. Thermus aquaticus. Remember. Next question. Can you do this question by your own? I will wait for 2 minutes. Can you do this? Observe the table in which size of different DNA fragments given and answer the question. Look at here. Fragments A, B, C. 700 kb and base pair, 700 base pairs, 150, 1500 base pair, 3000 base pair. In the process of separating DNA fragment, which fragment move faster? Come on, come on, come on. Any one of you? Any one of you? Rohan? Yes, yes. Think. Yes, gel electrophoresis. A move faster. Absolutely, I am correct. Absolutely correct. What we know about the gel electrophoresis? This is gel electrophoresis plate. Gel electrophoresis plate. Now we will load DNA at negative side, and this is the positive side. positive side. Now these are what, tell me, wells are there. In this wells we will add what? DNA. Our DNA is negatively chargeable, so we are adding what, tell me, DNA fragments here. Now DNA fragments, and we are connecting this to electrode, right? We are connecting to this, what, look at here, I am connecting like this. Right? Now answer my question. This DNA in the gel electrophoresis, this agarose gel, large DNA particles are slowly move. Large DNA particles are slowly move. Small DNA particles are further move. Yes or no? This is the thing we discussed, right? 700 base pair means small DNA. 1500 base pair means little big one. 3000 means bigger one. So I can write C is here, B is here, A will be faster. A will be faster. Am I clear? Am I clear or not? Yes, yes, answer me. Till here everything clear or not? Sir, how can you say gel electrophoresis depending on the separation of DNA based on size? Gel electrophoresis depends. What's the principle of gel electrophoresis, student? You should not forget this. Separation of DNA based on the fragment. So smaller fragment far away. Smaller fragment far away. Are you with me? You got my point? Clear? Understand my point? Yes, 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 yes. Understand my point? Right. Look at here. This process separates DNA fragment. Which fragment move faster? Which fragment? Smaller fragment move faster. Honest, answer me. How many of you got this question? How many of you understood this question? Come on, answer me. Show me your thumbs up. Till here everything, this question is clear or not? How many of you got this? Ayan, I need answer. Flip block, I need answer. Come on, come on. Rohan, till here everything clear? You got this point or not? You got this question or not? Yes, done. What about others? What about others? Right, sir, got it. Right. Yes, 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 yes. Next question. Come to the next question. This question tells us what this question is asking. Observe the diagram below. PBR 322. Answer the following question. Okay, complete PBR 322 the given. ROP, ORI, everything is given. Process of protein. Okay, now repress of protein. So answer the following. So what is PBR 322? Write the role of ROP, state the significance of ampicillin and also tetracycline. I am telling you, biotechnology chapter student, PBR diagram, PCR, gel electrophoresis 100% can come. Keep in mind. What is PBR? PBR 322 is a plasmid, is a vector, artificial prepared vector for the gene carrier, which contain 8 restriction sites, cloning sites, 2 selectable marker, 1 ORI, 1 ROP. Yes, yes, absolutely, it's a vector. Kitraj, correct. Now look at here. Write the role of ROP. Process of protein. ROP is that site which produce the protein essential for the replication of DNA. Process of what they call a protein. This ROP is the protein synthesis area which secrete essential enzyme for the replication of DNA or replication of this entire what process DNA vector. State the significance of ampicillin and tetracycline. Ampicillin, tetracyclines are the selectable marker. These two are helps to eliminate the non-recombinant and select the recombinants. Ampicillin, tetracycline. Next question. Name the given organism which vector showed in inserted to get a copy of a desirable gene. They are asking. Look at there, how many different type of questions are coming from the PBR 322. Name the organism in which the vector showed in the inserted. Sir, bacteria. We can insert this one into the bacteria. That is our transformer. Plasmid will be inserted into the bacteria. Am I clear? Am I clear? Right, Kitraj? Done? Now, here, next. Mention the area labeled in the vector responsible for the controlling the copy. Sir, ORI. Mention the role. ORI is the one which is essential for the making of copy, thousands of your desirable DNA copy. Explain and name and explain the role of selectable marker in the vector. Now all of you look at here. Insertional inactivation, one of the important selectable marker. All of you look at me. What I am teaching in the selectable, what they call only E. coli and bacteria. Yes, all of you pay attention and look at me. Insertional inactivation, how it work. This is bacteria I am taking. Look at here, I am taking, this is bacteria. This is E. coli bacteria. E. coli bacteria. In E. coli bacteria, this is what they call a plasmid. Sorry, this is own DNA is there. Directly I am writing what plasmid. This is plasmid. Okay, this is plasmid. I am taking this plasmid out. This plasmid has a gene we called LAG-Z gene. Look at here, this gene we are writing what? LAG-Z gene. LAG-Z gene code for, in molecular base of inheritance we learn, LAG-Z gene code for beta galactosidase enzyme. Yes. Now what I am doing, I am keeping this bacteria in synthetic lactose. Look at here, artificially synthetic lactose is there. This is normally white in color. White color. This is synthetic lactose. Synthetic lactose. Now I am keeping this bacteria in the synthetic lactose. Inducer, lactose is the inducer. So in presence of lactose, beta galactosidase enzyme, LAG-Z gene undergo transcription, translation to form a beta galactosidase enzyme. Now when beta galactosidase enzyme come, this enzyme convert white color lactose into blue color. Blue color. When medium convert into blue color, what does it mean? Beta galactosidase enzyme working, functional. Hold on. What I am teaching now, careful look at here. This is beta galactosidase enzyme. This is my desirable DNA. My target DNA. Look at here, flip flop, careful look at here. This is my target gene. When I am inserting my target gene in between the LAG-Z gene, this is my target gene. If my target gene has gone inside the LAG-Z gene will be inactivated. But I don't know that. It inactivated, my gene gone to the vector or not, I don't know. I inserted, I cut with the restructured endonuclease enzyme, I added the ligase enzyme, I joined this DNA particle. I don't know my gene is gone or not, I don't know. I don't know it is recombinant DNA or a non-recombinant, I don't know. I done this one. So what I'm doing, I'm sending back to the bacteria. I'm keeping this bacteria here where synthetic lactose. I'm keeping this bacteria in the synthetic lactose. Or you just keep this vector here. Am I clear? You just keep. What happened here? When your DNA has gone inside the LAG-Z gene inactivate. So if you keep here in the synthetic lactose, beta galactosidase enzyme will not come. When beta galactosidase enzyme not come, this synthetic lactose remain white color or colorless. If our synthetic lactose remain white color or colorless, what does it mean? The LAG-Z gene inactivated. That means your target gene has gone into the vector. You understand my point? You got my point? Till here everything clear? Kitraj, are you listening? Ayan, till here everything clear? When the medium is converting into a blue color, that means beta galactosidase enzyme is working. That means your gene did not inserted. When the beta galactosidase, when the medium is remain white color or colorless, our gene has gone inside. That means beta galactosidase enzyme inactivated. Good. Next one. Look at the next question. Write a Taq polymerase used instead of ordinary DNA polymerase in polymerase chain reaction. No, sir. We cannot use that ordinary DNA polymerase enzyme because ordinary DNA polymerase enzyme cannot be worked at 72 degrees Celsius. We discuss in biomolecule chapter 11th grade, when temperature increase, enzyme activity decrease. Taq polymerase enzyme can only tolerate that higher temperature. Name the source of or Taq polymerase enzyme come from the Thermus aquaticus. What is PCR used for? PCR used for the amplification of DNA. PCR also used for the early detection of diseases. Early detection of diseases also PCR use. Am I clear? Right, very good. Next question. Name the specific enzyme responsible for the nucleotide polymerization in DNA replication. DNA replication, name specific enzyme responsible for the nucleotide polymerization. I can simply write DNA, replicate to form DNA. The enzyme name is called DNA dependent DNA polymerase. What is the enzyme name? DNA dependent DNA polymerase is the enzyme which is polymerize the DNA from which direction to which direction? 5 prime toward the 3 prime. Write two characteristic feature of this enzyme. This is more accurate and this will be the one which is recognize the parental DNA and polymerize. More accurate one and polymerization takes place. The enzyme characteristic feature is it's a more accurate and also it polymerizes the DNA. And what they are asking, name the region of E. coli DNA where enzyme can initiate and replication. Enzyme replication, what they are asking, E. coli, name the region of E. coli DNA where this enzyme can initiate replication. ORI. ORI area where the replication takes place. ORI site. Am I clear? Okay, yeah, basically it increase. Yes. Next question. Why is coding sequence and enzyme beta galactosidase is a preferred selectable marker? Just now I explain you why beta galactosidase enzyme is a selectable marker. Explain why is a preferred selectable marker antibiotic resistant gene. Antibiotic resistant gene and Salaxi genes are selectable, preferred selectable marker because these two are inactivated when our gene gone. So we can easily identify with the medium color and also the growth of bacteria and ampicillin and also tetracycline. We can pick up our what they call transformer or rDNA. Let me take one small example. If you able to recall, recombinant cannot or transformants cannot grow in the tetracycline. Transformant can grow in the ampicillin is given. What is given in the inserity selectable marker? For example, this is our transformant. Okay, this is our vector. In the vector, this is our what is that? Our gene has gone. So if you keep this transformant, okay, transformant in tetracycline medium, it will die. The number will decrease. So stop. You are my transformer because the ability of resistant tetracycline you lost. How did you lose? Because of my gene. So this will not grow here. But if you take a non-transformer, if you take non-transformants can grow. They can grow in ampicillin as well as tetracycline. In both medium it is growing, non-transformer. Next, what are cloning sites of cloning vector? Cloning sites are the vector which recognize at which the restriction endonuclease enzyme act and cleave at a particular area. In PBR322, restriction sites are 8. Okay, explain their role. Name any such two sites are. Yes, I'll write here ECO R1. ECO R1 is one site. Okay, BAM H1. BAM H1, one more side. Explain their role. Exactly this restriction cloning site at which we cut the endonuclease enzyme and we insert our desirable DNA. Are you following me? Till here everything clear? So can you tell me about the selection of recombination in PBR322? PBR322 selection is tetracycline. Tetracycline in between, if you insert the gene and keeping the transformer in the tetracycline, the number will be decreased. Okay, the growth, the bacterial growth will be decreased, but the non-transformer can go in the ampicillin and also tetracycline both. If a bacteria is growing in both tetracycline and ampicillin, that is your non-transformer. Your transformer can grow in ampicillin, cannot grow in the tetracycline. Fix this one, Rohan Krishna. Next question, biotechnology application. Last chapter, biotechnology application. We will discuss RNAi, next artificial insulin, and also gene therapy. Look at the questions. What are the questions? What are the topic we should cover? Biotechnology and application. Biotechnological agriculture application, BT cotton. BT cotton is very easy. In this class, BT cotton I will not discuss because BT cotton everyone knows. Cry protein, cry genes, everyone know this one. We will go with what? BT cotton. We will go with RNAi interference, biotechnological applications in medicine, genetically engineering insulin, especially gene therapy, ADA enzyme we will write, ADA enzyme, molecular diagnosis, PCR, and also ELISA, enzyme linked immunosorbent assay. Next, transgenic animals. These are the one we will cover. Look at the first question. This type of question will come in the exam. This is artificially produced insulin from the bacteria, E. coli bacteria, Eli Lilly company. Look at here. Refer the diagram and measure pro-insulin to insulin. Answer the following question. Fine. This is pro-insulin diagram. B peptide, B insulin, B peptide, A polypeptide, everything is given. C polypeptide also given. What is the first question? How are two short polypeptide chains are insulin linked together? Any one of you answer this one? How they are linked together? How they link to sir disulfide bonds? Between the two chain, disulfide bond bridge is there. Absolutely correct. Here, state the role of C polypeptide in human insulin. C polypeptide role is connect the AB polypeptide. When C polypeptide is there, it's called the inactivated insulin called proinsulin. Next, mention the chemical change that proinsulin undergo. It can be able to act as a mature insulin. The chemical change is the C polypeptide chain will be removed so that AB polypeptide chain between disulfide bond form, it will become a mature insulin. My dear children, insulin topic carefully prepare and go. I'm really telling you, insulin very, very important. Very, very important. Next, list the advantages of insulin obtained from the pancreas of sludged cows and pigs. List the disadvantages. Sir, this type of insulin is allergic to the people. This type of insulin is allergic to the people. Instead of curing that one, instead of the diabetic curing, they are feeling that allergy developing. So, biotechnology decided to take the human gene and insert into the bacteria and get the human insulin. Got my point? So this is the first 5-mark question. If this type of simple question come, 100 out of 100 our students will get sure, right? I hope you all are. Next, gene therapy. Gene therapy is an attempt to correct the genetic defect by providing a normal gene into an individual, right? Into individual. By this, the normal function can be restored. Alternate method would be provided the gene product, protein enzyme, known enzyme replacement therapy, which would also restore the function. Fine. Gene therapy, ADA enzyme therapy, thoroughly prepare. Which is your opinion is better option? Give reason for your answer. Now, in gene therapy, attempt to correct the genetic defect by providing normal gene into an individual. Fine. By this, normal functioning can be restored. Alternate method would be the providing gene product, protein enzyme, known to be an enzyme replacement therapy. Which is your preferable method? Any one of you? Yes. Absolutely. Very good, Ayan. Absolutely good. Gene transfer method, sir. Gene therapy. We can give a permanent cure. Enzyme replacement therapy, bone marrow replacement therapy will not give a permanent cure. You have to repeat that one for the periodically. So it is a high expensive process. So if you do gene therapy in early embryonic stage, permanent cure. Absolutely correct answer. A person is born with a hereditary disease suggests the possible corrective method for it. Illustrate by giving a specific example. A person born with this specific genetical disorder, gene therapy is a good option to identify the disease at early embryonic stage. Not early embryonic stage identification. We will go with the pedigree. We will go with the family background, and after that we will see. And this particular baby is going to this embryonic stage only we will do the gene therapy, so it will give a permanent cure. Permanent cure. ADA enzyme therapy questions can come in different manner, students. Remember. Next, name nematode that infest and damage tobacco roots. The nematode name is Meladogyn incognatia. Affect the tobacco root. To kill that Meladogyn incognatia, we are using the mechanism. How are transgenic tobacco plants products are solve this problem? This problem is solving by a simple mechanism called RNA-I interference mechanism. This RNA-I interference mechanism is silencing the mRNA of this nematoda, so that essential protein is not translating in the nematoda, so the nematoda cannot grow. RNA-I mechanism. Meladogyn incognatia worm reading. That worm, we are going to be read that worm with the help of RNA-I interference mechanism. If a desired gene is identified in an organism for some experiments, explain the process of following. Cutting the desired gene at specific location. Synthesis of multiple copies of this desired gene. They are asking. If desired gene is identified organism for some experiments, explain the process of the following. Cutting the desirable gene. Cutting the desirable gene with the help of a specific molecular scissors called restriction endonuclease enzyme. With the help of restriction endonuclease enzyme, cutting is possible at a desirable area. So insertion is done by the DNA ligase enzyme. Look at here. Synthesis of multiple copies of desirable gene is possible if you select the vector. That vector have a ORI site. So when you insert the DNA into the vector will be multiplied many times. So your desirable DNA will become cloned. So multiple copy. Done. Simple question. Done, Ayan? Done, beta? Right, very good. Next one is, write the help of diagram shows different steps of formation of recombinant DNA. 5 mark question. I told you rDNA technology, two type of questions can come. One is the step-by-step process, one is diagrammatic process. I am not drawing here, there is no time. Prepare this question thoroughly at home. Diagrammatic preparation and also step-by-step preparation. Now what they are asking, restriction endonuclease enzyme eco-R1. Name the technique that is used to separate the fragments of DNA DNA cut by the restriction endonuclease enzyme. Gel electrophoresis. Ethidium bromide we are adding and we are exposed to UV rays. Orange bands are forming. We are taking that, we are isolating the orange band. We call the illusion process. Illusion process. Done. You got my point. So we will write what? Gel electrophoresis process. Next question. These 3-4 questions are very important question, and the last question. I think many questions are there. I think so. Chalo, we'll see. Name any 2 techniques that serve the purpose of early diagnosis of some viral diseases. I can write PCR and enzyme linked immunosorbent assay. These 2 are identifying the disease at early embryo at early stage. Without symptom formation only we can identify the diseases. How does silencing of specific mRNA RNA interference preventing the parasite infestation? We are introducing the nematodal gene into the crop, transgenic crop, to nematoda, tobacco, with the help of bacteria, like Agrobacterium tumefacium. In the plant body, this DNA, look at here, look, this is nematoda, this is a nematodal worm. This nematodal worm DNA we are introducing into the plant. So this plant will become what? Transgenic plant. Remember, this is a transgenic tobacco, right? In the transgenic tobacco cell, look at it, this is a cell. Here I am writing, this is big cell I am taking here. In the cell, the viral DNA undergo, both strands undergo transcription. So form double-stranded RNA, dsRNA. dsRNA will be spliced, they cut to form a small RNAs. This small RNA is called siRNA. Am I clear? siRNA. When nematoda come and eat, the plant part, it will go into the nematoda body. When nematoda trying to transcribe the mRNA and translating to protein, this siRNAs are complement to this mRNA, so that mRNA not going to be translate in the nematoda worm. That's how RNAi mechanism work, and it is not allowing mRNA to translate. This is a big mechanism, actually you should learn in school. This is a big mechanism. I don't have time to explain the entire mechanism. If a time is there, I would have explained this RNAi beautiful manner, because the big process it is. You should understand the first normal RNA interference in us, and after that, once RNA, double-stranded RNA virus affect us, what exactly happened, how our body protect, that you must know. So only in brief I explain this one. Next, explain the name of enzyme ADA, adenosine deaminase. Why the enzyme is essential in human body? This enzyme is secreted from the BT lymphocyte cells. This enzyme is essential for the maturation of BT lymphocyte cells. BT lymphocyte cells are acquired immunity, very important immune cells. Suggest gene therapy for this deficiency. For the deficiency of ADA, gene therapy is the best suggestion. In the gene therapy at early embryonic stage, or they collect the patient blood. That patient blood is immature lymphocyte. They culture in vitro with the help of retrovirus. They inject the desirable gene into these lymphocytes, and lymphocyte will be back to a patient blood. Periodically it should be happen. If you wanted to get the permanent solution, you should do this gene therapy at early embryonic stage. I am telling you, student, biotechnology application, PBR3 to biotechnology application, PCR, gene therapy, insulin, and also what they call RNA interference mechanism, transgenic animal. This 5 topic thoroughly go, 100% question can come, no doubt. 100% question come. How did Eli Lilly synthesize the human insulin? Human insulin is prepared by the American company Eli Lilly. With the help of genetic engineering, they isolate the DNA, they introduce into the bacteria, plasmid and vector, and bioreactor. They grown this bacteria in the bioreactor, and downstream process they obtain what? Product. Mention one difference between the insulin and one produced by human pancreas. Eli Lilly company producing direct mature insulin in which there is no C polypeptide chain. In case of us, natural human insulin produced in the form of proinsulin first, which contain A polypeptide, C polypeptide, B polypeptide. Later it processed to form a matured insulin, which C polypeptide will remove. But in case of Eli Lilly company, what it is doing? Only AB polypeptide is a mature insulin it producing. Are you following me? Are you there? Are you following me? Right. Last questions. Look at here. One more question. 3 more questions are there. We will finish. And look at here. All of you. Let me finish this one. What is plasmid? Plasmids are self-replicable extracircular double-stranded DNA. What is mean by ADA deficiency? Due to the deletion of gene, ADA enzyme unable to synthesis in the blood, in the cells, RBs, what they call T lymphocytes and B lymphocyte. That deficiency, that result to the formation of a acquired immunity loss. That acquired immunity loss deficiency called ADA deficiency or SCID, SCID disease, severe combined immunological deficiency, SCID. Now, how is gene therapy is solution is problem? We discussed this one. Permanent cure is early embryonic stage. Can any one of you answer this question? How Rosie considered the different from the normal cow? How Rosie different from the normal cow? Any one of you? Rohan, how it is normal? Sir, Rosie is a transgenic cow. Human mother milk protein gene has been introduced in the Rosie. So the Rosie cow is producing alpha lactalbumin human protein 2.4 milligram in 1 liter. So normal cow is not producing the human alpha lactalbumin protein, but Rosie does. So our human gene introduced into Rosie. So Rosie is a transgenic cow. This you will get in biological product. The useful of what? Transgenic animal. What is ADA? We already discussed this one. 3 methods of cure. 3 methods of cure: enzyme replacement therapy, bone marrow replacement therapy, gene therapy. Right? These are the important questions from 4 chapters, student. Thank you very much for attending this session. And moreover, remember all of you, properly prepare and go. After my class, take your dinner and come back to books again. Prepare 100 out of 100. And moreover, whatever the questions, whatever the topic we skip or we miss here, also cover. And all the very best. I cover maximum chapters, maximum important chapters I cover. Last chapters, ecosystem, organism, population, I did not. Tomorrow morning I'm planning, but one day should be there. One day should be there to prepare yourself. That's why important chapters only I covered today and yesterday. Make clear? All the very best. Leave your fear, leave your complete what they call fear, everything at home. Peacefully go to exam hall. Try to read the question once and twice and attend. Make clear? Try to maintain the answer sheet very neatly. When diagram comes, properly label, properly write. All the very best for your final examination. I hope it will come very good paper and you will get good marks. Thanks for attending Brilliant All classes. Thank you. Good night all of you.
Brilliant Qatar, your trusted coaching partner for IIT, JEE, NEET, Science and Commerce tuitions with 10 years of excellence in quality training. Brilliant Qatar.